Good morning, everyone. Good morning, everyone. May I request all the guests to be seated so that we can begin the proceeding shortly, please. Good morning to all the distinguished guests. We welcome you to the third Dr. Anji Reddy Memorial Lecture, an event dedicated to honoring the profound vision and contributions of Dr. Kallam Anji Reddy. The vision of our founder, Chairman Dr. Anji Reddy, recipient of Padma Bhushan in 2011, can be articulated in his own words. He said, "If the world's burden of disease is to be diminished, it needs science that is both good and cost-effective. I have great confidence in the army of chemists and biologists and firmly believe that working on the trial of innovation will lead to creating a great company, a company that doctors, patients, investors, and the public will admire. We at Dr. Reddy's are walking his path of vision and innovation." Thank you everyone once again for joining this event in person, and thank you to all those joining us online. Before we begin, may I request Security in Charge, Mr. Javed from Awasa, to brief us about the safety protocols, please.
Good morning, everyone. My name is Jabir. I am the Security Manager for Awasa. I am here to brief you about security precaution. Currently, we are at level 1, Constellation Hall. Nearest fire exit located nearby. All the fire exits we fixed up. For identification, we fix signages. In case of any emergency, please use nearest fire exit to reach the safe assembly point, which is near to the main entrance. And the hall is purely designed with all the safety and security equipments. In case of any fault alarm, we have our disaster management team to handle the situation. Don't panic. In case of emergency, please, we have our team here to serve you. Please inform them. They will connect with the disaster management team. Thank you.
Thank you, Mr. Javed. I now request Mrs. Anuradha, Mrs. Deepthi, Mr. Satish Reddy, Mr. G.V. Prasad, and Ms. Malika to please come onto the dais and light the lamp. I also invite Mrs. Ranjani Shivashankar for the invocation.
सामजवरगमना साधुहृत् सामजवरगमना साधुहृत् सामजवरगमना साधुहृत् सामजवरगमना साधुहृत् सारसाब्जपालकाला तीतविख्यात सामजवरगमना साधुहृत् सारसाब्जपालकाला तीतविख्यात सामजवरगमना सामनी गमज सुधा मयगान सामनी गमज सुधा मयगान सामनी गमज सुधा मयगान विचक्षण गुण शीलदयालवालमा पालय सामजवरगमना वेदशिरो मातृज सप्तस्वर नादाचलदीप स्वीकृत वेदशिरो मातृज सप्तस्वर नादाचलदीप स्वीकृत यादव कुलमुरळी यादव कुलमुरळी यादव कुलमुरळी वादन विचक्षण गुणशील दयालवालमा यादव कुलमुरळी वादन विनोद मोहन करत्यागराजवन्दनीय सामजवरगमना साधुहृत् सामजवरगमना साधुहृत् सारसाब्जपालकाला तीतविख्यात सामजवरगमना
Thank you, everyone, and thank you, Mrs. Ranjani. I would like to also specially thank Mrs. Anuradha for her guidance in planning this event. Thank you, ma'am. We all know that Dr. Anji Reddy was a distinguished scientist himself, besides being a philanthropist and entrepreneur. We'd like to present a video as a suitable tribute to his remarkable journey. Can we have the video, please?
In electricity only when I was 12 years old or something. It was a dream. it was not a plan on the drawing board, no.
No.
But it was a dream. I want to be a pharmaceutical manufacturer. And obviously, God willing, maybe number one in India. If I didn't have a vision at various stages in the evolution of this company wouldn't have reached this stage. You should not think that because of failure we should cut corners or anything like that. The vision should continue in spite of the failure, because if there is that element in you which is better than many other people, you can be one of the top companies. This foundation kept about the poverty of India, and he wrote a sentence which hurt me so much. He said, except that certain regions of India are worse than sub-Saharan Africa. I was so angry. What really drives me is the science. That can I finally bring a drug into the market which can change the way people live?
Thank you, team, for a captivating video. I would now request Mr. G.V. Prasad, Co-Chairman and Managing Director, Dr. Reddy's Laboratories, to deliver the welcome address, please.
Thank you. Good morning and a warm welcome to all of you. I was just imagining if Dr. Reddy was in this room, he would have been really proud of all of you, your friends, scientists, colleagues, and be part of this great event. I'm sure he's somewhere there watching and smiling. Thank you for joining us today for this third Dr. Anji Reddy Memorial Lecture. A warm welcome to everyone in the room as well as those who have joined us online through the streaming link. Let me begin by sharing some sad news. This year, on February 19th, we lost Mrs. Samrajyam, Dr. Anji Reddy's better half, my mother-in-law. And she was a pillar of support to Dr. Reddy in his journey, and of course to all of us. She was well known for her service to the less fortunate through various activities as part of several philanthropic organizations. She touched thousands of lives and was well known for her kindness, approachability, and relationships across the societal spectrum. We will miss her presence. So may I request 2 minutes of silence to pay respects to her memory. Thank you. Thank you. So as most of you here know, Dr. Reddy was dedicated to advancing science in the pharmaceutical industry, and he pursued science and technology through his relentless efforts with a focus on affordability, access, and innovation. And this event is to celebrate his passion for science and technology. We are honored to welcome Dr. Ashok Venkatraman, a very distinguished cancer biologist whose research has contributed fundamentally to the current understanding of how cancer is suppressed by genes that maintain the integrity of the human DNA. And these genetic mechanisms are very pertinent to understand how cancers manifest, in particular breast cancer. And his work has opened multiple avenues for the identification of new druggable targets to find cures for cancer. And many of you might be familiar with his pioneering discovery of the mutations in the BRCA gene, a key cause for breast cancer. And this has helped people susceptible to breast cancer for early prognosis and monitoring and treatment. So Dr. Venkatraman's efforts also to create networks across Cambridge, UK, Singapore, and India is going to see high impact on the cancer research. And there are many things I think that his work the best of his work is still to come. And we look forward to his lecture today. I'm also pleased to welcome Dr. G. Nageshwar Rao, founder and chairman emeritus of L.V. Prasad Eye Institute. Dr. Rao, or Nag as he asks us to address him, is a healthcare icon whom Dr. Reddy admired immensely for his disciplined approach to institution building, advancing science, and serving patients from all sections of society. Dr. Rao's commitment to delivering the highest quality eye care, adoption of cutting-edge technology, and focus on research is quite legendary. The inspiring byline for the institution he founded is, "So that all may see." And the institute that he founded does all this completely free to people who don't have the means. Beyond treatment, he has also built a lasting organization which he has set up to continue flourishing and grow even as he steps down from day-to-day leadership. Today, LVPI is recognized as one of the top 10 eye care institutions in the world, touching millions of patients world over. A Padma Shri awardee, Dr. Reddy has been inducted into the Ophthalmology Hall of Fame at the meeting of the American Society of Cataract and Refractive Surgery. He has served as a board member, Secretary General, and President of the International Agency for the Prevention of Blindness. And today we hope to learn from some of his experiences with Dr. Reddy. Excuse me. Deep science was at the heart of Dr. Reddy's vision for affordable and accessible medicine. He had this drive to be a pioneer in science, to be a pioneer in business. And this hunger for being the first stemmed from his scientific training. As you know, in science, there's always this burning desire to do something novel that has not been done before, and he brought the same philosophy to business. While money mattered, I think it mattered more for him to be the first to market with products that may have an impact and a positive impact on as many people as possible. Dr. Reddy left a deep mark on many of us who interacted with him, and I learned from him how to explore the magic of science, like many of my colleagues have, how to think without limits, and to mobilize people towards shared goals that matter for society. He was passionate about using science and innovation to find solutions for society. He wanted to find solutions for serious unmet medical needs, as you just saw in the video. And he invested relentlessly in science and technology with a focus to make the world a better place for everyone. Of course, making medicines affordable and accessible to patients drove him in his initial phase of business life. He was not concerned too much about limitations, and in a way he was supremely self-confident. You could see that in his talk in the video. He was not worried about resources, and he used to say that resources would become available if he pursued the right vision. And he had pioneered many technologies and helped jumpstart the pharmaceutical ecosystem in India, and helped India in being recognized as the pharmacy to the world. And the industry recognizes his role in this whole effort. Dr. Reddy also encouraged us to take care of the planet, people, and patients. While sustainability was not a major focus for him personally, he pushed us in taking a broader perspective in everything we did. And we took up sustainability as a key pillar of the company based on this tenet. And today we have many accomplishments in the area of sustainability, and we are counted as one of the best performers in this space across the world. These facets of life, which have inspired the title of this lecture series, Science, Society, and Sustainability, are also embedded in the culture of the organization that he founded. Our achievements at Dr. Reddy's in developing complex products, first time in India, breaking into new and difficult markets, new drug discovery programs, especially in cancer now, and our investments in emerging areas of science and technology like CAR-T, cell and gene therapies, bispecifics, are all based on the scientific ambition he instilled in us. And we want to use this lecture series to inspire people to pursue science for society and sustainability. And we hope this event will promote science as a way of moving forward for the benefit of all. Today's guests represent the extremely exciting scientific developments that continue to take place around the world. Nothing would have given him more pleasure than being among his fellow scientists and forging such connections between industry, academia, and research institutions. And we hope to bring world-renowned scientists to India through this event to encourage the pursuit of science and technology in the broader ecosystem. And this is our way of paying tribute to the scientist, entrepreneur, and philanthropist who Dr. Reddy was. With that, I'd like to welcome and thank Dr. Ashok Venkatraman, Dr. Nageshwar Rao, and everyone else assembled in this room for your presence at this third Anji Reddy Memorial Lecture. Your encouragement will help us inspire many scientists to make this a reality, his dream a reality. Thank you very much.
Thank you, Mr. Prasad, for the welcome and your thoughts. We'd like to now present glimpses of the past editions of Dr. Anji Reddy Memorial Lecture, established on the themes of science, society, sustainability. It was launched in 2023 to pay tribute to our founder, Dr. Kallam Anji Reddy's passion for deep science and his dedication to profound scientific acumen and integrity. The event has gathered prominent scientists from both industry and academia, attracting notable personalities from the scientific community, including Nobel laureates. Can we play the video, please?
Can I finally bring a drug into the market which can change the way people live?
Thank you. And moving forward, I would like to invite Dr. Srinivas Oruganti, Director of Dr. Reddy's Institute of Life Sciences Drills, to introduce our speaker for the memorial lecture, Professor Ashok Venkatraman. Dr. Oruganti, please.
It is indeed my privilege to invite and introduce Professor Ashok Venkatraman. We are indeed grateful for having accepted to deliver the third Dr. Kallam Anji Reddy Memorial Lecture. Professor Venkatraman is the Director of Cancer Science Institute of Singapore. He is also a distinguished professor of medicine at the National University of Singapore. He also serves as a research director at the Institute of Molecular and Cell Biology in Singapore and the executive director of NRF's National Initiative for RNA Biology and its applications. Professor Venkatraman received his medical degree from CMC Vellore and then went on to pursue his PhD at University College London. He joined the Medical Research Council Laboratory of Molecular Biology in Cambridge University, and he was selected to be the first holder of the very prestigious Ursula Zollner Professorship of Cancer Research at the University of Cambridge. From 2006 to 2019, he was the director of the Medical Research Council Cancer Unit at Cambridge University. Professor Venkatraman was elected a Fellow of the Academy of Medical Sciences, London in 2001. He was also appointed as a member of EMBO European Academy Heidelberg in 2004. As Mr. Prasad has mentioned during his welcome address, he is widely recognized for his contributions to understanding the genetics and biology of cancer, particularly in elucidating inherited cancer syndromes which are associated with genomic instability and discovery of mutations in the breast and ovarian cancer gene BRCA2, we call it as BRCA2. He received the very prestigious 2017 Basser Global Prize for his pioneering work on the BRCA tumor gene and mutations. In addition to his very bright academic career, Professor Venkatraman also has the hallmark of being an entrepreneur, a hallmark of his entrepreneurial spirit. Professor Venkatraman is a co-founder of Foremost Limited. It's a biopharma company focused on a very proprietary platform called SiteSeeker. It's a protein interference platform for phenotypic screening in drug discovery. He continues to contribute his time and effort to promote biomedical research in India. I was personally fortunate to get connected to Professor Venkatraman in 2011 when the very ambitious consortia of drug discovery across all the units, multiple units in India was being conceptualized. Professor Venkatraman is a collaborative science chair at INSTEM Bangalore and NCBS Bangalore. He coordinates the Center for Chemical Biology and Therapeutics, known as CCBT, in Bangalore. Professor Venkatraman is also the coordinator and Metha Distinguished Chair at the Center for Integrative Biology and Systems Medicine at IIT Madras. With this brief introduction, I have a few more points to cover, but we will all be waiting to listen to you. It's indeed a privilege, Professor Venkatraman, and we look forward to your lecture and understanding the shared legacy and passion for drug discovery and improving human health. Thank you.
Thank you very much. Yep, my lapel mic is on, yes. Yeah. I'd like to actually start by thanking everyone, particularly the Reddy family, and remember why we are all gathered here. Dr. Anji Reddy has been a great inspiration, as we've heard, and it's a great honor for me to be standing here in front of all of you to deliver a lecture this morning. So I'm very honored and also very humbled to follow in the footsteps of the many eminent scientists who have spoken at this meeting before, and I extend once again my great thanks to Dr. Reddy's family for having invited me to do this. I'm going to be speaking today about my work over the last 2 decades or so on understanding and suppressing the genesis of cancer. This work started in Cambridge in my laboratory at the Medical Research Council Cancer Unit, and it's continued after my move about 4 years ago to Singapore. So it's really going to be a sort of scientific journey that I'd like to take you through. I understand that this is a very mixed audience, so I hope that the aficionados and experts in the audience will forgive me for trying to reduce some rather complex concepts to simple terms. So please forgive me, but I will answer your questions later if there's time, in case you want to understand details. So cancer, you know, starts in lesions that involve just tens or thousands of cells. But it grows from there to lesions that involve billions of cells. We know that at very early stages, cancer is potentially curable, not just treatable, but potentially curable. But the sad fact is, cancer is usually treated when it is much more advanced. And despite the fact that we have known for over 30 years that early intervention in cancer can greatly improve clinical outcomes, less than 20% of cancer research worldwide focuses on this really, really key issue. When I talk about early intervention in cancer, as in any chronic disease, please be aware that the scope is really quite vast. Some ideas are sketched out for you here, and I'll walk you through some of the issues that are highlighted. So we all know that cancer is primarily a disease that affects us as we get older, and that it incubates within us without causing any symptoms for a long, long time, often decades. Clearly, there is a lot that we can do to prevent cancer, primary prevention it's called, before it gets a foothold and before it progresses. For example, we have all heard and seen examples of how changes in behavior or lifestyle, including diet, can help to reduce the risk of cancer. Avoiding exposure to toxins in the environment. And very interestingly and importantly, we are beginning to see the advent of the use of drugs, not to treat chronic diseases, but potentially to prevent chronic diseases like cancer. Cancer is diagnosed, sadly, in many countries, in many instances, when it's already advanced. And if only we could diagnose later stage cancers earlier, the success in treating them would be so much higher. So how does one go about this? Again, I think many of you will have seen and heard about interventions like mammography in women who are postmenopausal, in using low-dose computed tomography in high-risk groups for lung cancer, for example, people who smoke. And these all bring value because you can diagnose and treat the disease much earlier. Really interestingly, in recent years, the potential to diagnose cancer before it becomes clinically evident has been manifest. For example, companies like Grail have developed tests which are based on blood, which can detect circulating alterations in our DNA, which come from tumors years before they may manifest. When we get to treating cancer, again, there are elements of early intervention. Cancer is very difficult to treat, and after primary therapy using any of these modalities, often it progresses or recurs. So another aspect of early intervention is detecting and intervening early after primary therapy, in other words, to prevent or potentially to early detect instances of progression or recurrence. And what's emerging now, particularly relevant to the pharmaceutical industry, is that as we get safer and better tolerated drugs, which are based on deeper understanding of mechanisms, some of these agents may actually have use in chemoprevention. None of this, I submit to you, is possible without understanding in more detail how cancer arises. And essentially, I like to view cancer and carcinogenesis as a battle between two competing forces. One, what we call oncogenic influences, the name says what it means. They promote carcinogenesis. They make cells divide in our body without controls. And at the same time, tumor suppressive influences, which prevent oncogenic forces from getting out of hand and prevent cancer from arising. And it's when both of these break down that we actually progress from oncogenic influences, the breakdown of tumor suppression, towards the genesis of cancer. And without understanding in great detail how these steps actually occur in us, it's almost impossible to have a science-based strategy for early intervention in cancer, or that said, almost any chronic disease. So what I'd like to take you through now, with that by way of introduction, is some of our recent work on this set of ideas, which I hope will start with an axiom, really, that I think we're all familiar with. What is it that causes a risk of any disease, including cancer? It's our genes, which we inherit from our parents through generations, coupled to our environment, what we expose ourselves to, what we eat, what we breathe. And together, nature and nurture determine cancer risk. We still do not understand how this works, and I'd like to share with you some vignettes from our recent work on some of the mechanisms that may underlie this intersection. So again, for this mixed audience, let me take you through a brief summary of some of the ideas that I'd like to talk about today. I think all of you will be aware that fundamentally, the information that makes us what we are is encoded in our DNA. I like to think of DNA as a book with many, many billions of letters written in chapters. Not all of these chapters are read in every cell in our body. A cell in the liver may read certain chapters of this book. A cell in the eye may read other chapters. Same book, different cells. And that is because the DNA encodes molecules like proteins, which make the cells what they are. And these cells come together to form organs which make us what we are. And we are increasingly being able to understand how DNA or genes contribute to diseases because of the advent of many new technologies that have enabled us to very rapidly and cheaply read this huge book through sequencing of DNA. What's interesting about this book of life is that it is not static. Every time a cell divides, the book has to be copied. And it has to be copied perfectly without any errors, or the progeny will be sick. So the copying of DNA requires a vast machinery of proteins that enable this to occur without errors. But errors do occur, not just because this process is not perfect, but also because we're exposed all the time to lesions in our DNA. If you stand outside in bright sunlight for an hour and you look at 1 square millimeter of your skin, the ultraviolet light will induce thousands of alterations in the DNA. Why don't we all get cancer every day? It's because there is a fantastic machinery within our cells that repairs these lesions. Repairs, for example, the ultraviolet light-induced damage. Repairs, for example, errors during DNA replication. And also can repair a really bad problem, which is breakage of DNA, where one or both of the strands of DNA may be cut asunder, which really leads to serious problems. So in other words, our cells contain this machinery that is constantly accurately copying and repairing DNA when it needs to be repaired to maintain our health and well-being. Over hundreds of years, it's become clear that if we cannot maintain this book of life accurately, and the genome, the DNA, becomes unstable, this is connected with cancer. The observations go back to the 1890s when a pathologist, von Hansemann, could see the DNA in cellular chromosomes behaving abnormally in cells that he thought were destined to become cancer cells. Theodor Braun-Boveri made similar observations. And there are rare diseases like Fanconi anemia and Bloom syndrome where the DNA is clearly broken, fragmented, abnormal, which are connected with many problems, skeletal malformations, developmental defects, but also with the early onset of cancer. So it's clear that if we cannot maintain our DNA, there's a higher risk of developing cancer. And that is where our work began to come to fruition about 20 years ago, when the first genes, alterations in which caused common cancers like breast cancer, ovarian cancer at a high risk, were cloned by multinational efforts across the UK, the United States, and many other countries. And these efforts focused on familial breast cancer, women who have a very, very high risk of breast cancer running in the families. And two genes were cloned, the breast cancer genes BRCA1 and BRCA2. And although they cause only a small number of cases of breast cancer, they do so with extremely high risk. So if you inherit a defective copy of one of these genes, you have a very, very high risk of developing cancer. Not just breast and ovarian cancer, but indeed for BRCA2 in particular, a high risk of cancers of the pancreas, prostate, and many, many other organs. So when I was starting my laboratory in Cambridge at that time, we wondered whether understanding how alterations in these genes cause an excess cancer risk would teach us important lessons about the origins of cancer. BRCA1 and BRCA2 have become rather famous now, not through any efforts of the scientific community, but because a very prominent Hollywood actress, Angelina Jolie, is one of the many, many people across the world whose family has inherited a mutation affecting BRCA1. She's had to undergo prophylactic surgery after she's completed her family to reduce the risk of ovarian and breast cancer. It's just a symbol of how this disease affects so many women and men across the world by causing an excess risk of breast cancer, as you can see here, comparing between normal versus people who carry mutations in BRCA2 or BRCA1. So as I said, BRCA1 and BRCA2 were cloned in the mid-1990s by international consortia, and we were very fortunate, as I was starting my laboratory in Cambridge, to create a model using genetically engineered mice to ask how these genes might function. And in that mouse model, where we were recapitulating what happens in humans who have mutations in BRCA2, we found this really striking observation. And these observations were actually made by Ketan Patel, who was at that time a postdoctoral fellow in my lab. He's now the director of the Weatherall Institute of Molecular Medicine at Oxford, where he has continued a lot of this work. But Ketan and I made the observation that in cells which lack BRCA2, I hope you can see it. This is not normal, right? These chromosomes in which DNA is packed are broken, twisted, and you can see some of the aberrations highlighted for you here. And this was very exciting to us because it taught us for the first time that what BRCA2 was doing to prevent cancer was actually to make sure that DNA was repaired and the book of life was maintained intact in each of our cells. Now some details. My work over the last 20-odd years since that discovery has been first to understand in molecular, really atomic resolution, what BRCA2 actually does to repair damaged DNA. And for the experts in the audience, I will just say we have discovered it has two functions. One, I told you that DNA can be broken by many lesions that we encounter in our environment. And one function of BRCA2 is to enable that DNA to be repaired without errors. All right? So BRCA2 is essential for error-free repair of double-stranded DNA breaks, and that process is called homologous recombination, or HR for short. Second, DNA is being copied all the time. And we know that when the copying machinery is running across DNA, sometimes it stops. And when it stops, BRCA2 comes in and helps to restart DNA copying, and that's its second function in DNA replication, or what we call replication fork protection, or RFP for short. And we've been able to show over the last many years how this process works at atomic resolution. So we have been able, in the test tube, to reconstitute all of the steps, the molecules, the proteins, the DNA involved in homologous recombination and replication fork protection, and show that what BRCA2 does is to influence this enzyme called RAD51. And this little enzyme is present in virtually all life forms, from the simplest bacteria to us. Why? Because all organisms have to be able to copy their DNA. All organisms have to be able to repair broken DNA. So this molecule goes back to the origins of life itself. And what BRCA2, the breast cancer gene, does is to control how this molecule binds to different forms of DNA, either double-stranded or single-stranded, in order to enable efficient repair. And over the years, my laboratory has been able to show how this works to solve, using X-ray crystallography, the structural basis for these functions, and more recently, using cryo-electron microscopy, as well as single molecule resolution microscopy, to show how this works in cells. So I think we understand this rather well, but that's not what I'm going to be talking about today. What I'm going to be talking about is what comes afterwards, which is how do alterations in BRCA2 enhance our cancer risk? How do these alterations cause cancer? And I have to remind you that, as I said before, genome instability, inability to accurately repair our DNA, is a key cause of cancer. We know this now because we know that as cancers progress from normal cells to cancer cells, the instability of the genome, of our DNA, increases. And this has become evident through enormous progress in our ability to read DNA through the methods of next-generation sequencing. The first cancer genome in 2005, tens of thousands of cancer genomes now available, which are now moving from research to guiding therapy in the clinic. You might be forgiven if you had listened to me so far for thinking, oh, it's all in our genes, but it's not. We've known for hundreds of years that the origins of cancer are not just in our genes. Percival Potts in London in Victorian times found that chimney sweeps who were exposed to coal tar dust were developing cancers of the skin and scrotum through this exposure. And he showed that removing that exposure could decrease the incidence of cancer. And as you know, Dahl and Hill in the 1950s clearly showed that smoking, another environmental exposure, enhances the risk of lung cancer. And this just vividly demonstrates how the environment, our lifestyle, our habits contributes to cancer. You can see over time the per capita cigarette consumption and the correlation with lung cancer death rates in males in blue and females in red. I hope you can see that after these seminal studies reported by Dahl and Hill in Oxford in the 1950s, when we became aware of that link, and despite the best efforts of the tobacco companies, people did start to decline the habit of smoking, and you can see how that immediately, within years, began to impact the lung cancer rate. So this is a wonderful example of how it's not just genes, but our environment also that influences the genesis of cancer. What is our modern conception of the way that cancer originates? This is really stunning, and sometimes I have difficulty in comprehending this myself because it's become clear over the last 5 to 10 years through DNA sequencing. What my colleagues in the Sanger Institute in Cambridge in particular, and other colleagues across the world have been doing, is just to look at normal tissues, normal skin, normal organs, and ask, in these normal tissues, do we see any of the mutations, any of the changes in DNA that are connected with cancer? What would you guess? If you'd asked me, I'd have guessed not often, but that's not true. What we now know is that cancer-driving mutations, hidden assassins, I call them, are present in apparently normal cells, in apparently normal tissues, in all of our bodies, and they accumulate as we age, which is probably why cancer is primarily a disease of old age. And again, it's becoming clear that if you inherit a poor genetic background, you can have an increase in these mutations. If you inherit a positive genetic background, you may reduce the occurrence of these pre-malignant cells. And we think that environmental exposures may also influence the frequency of these pre-malignant cells. Now, if these cells are present in all tissues across our body, again, why don't all of us develop cancer? And that's because, again, as I said before, there are two competing forces: the oncogenic driving mutations and the tumor suppressive mechanisms in our bodies, in our cells, that prevent these from becoming cancer. And only when both of these break down do you actually get the disease. How do tumor suppressor genes work? These are genes whose job it is to prevent early cancer cells from becoming cancer. We'll call them TSGs, tumor suppressor genes for short. You know, most genes in our body, we have two copies. One copy from our mother, one copy from our father. These tumor suppressor genes, sometimes we can get a bad copy, a faulty copy, like you can of BRCA2 from one of your parents. Yes? But you still have one good copy inherited from the other parent, shown in green. Al Knudsen, working in the 1970s, showed, studying eye cancer, retinoblastoma, a rare form of childhood eye cancer, that when you get cancer, both copies have to be inactivated. It's called Knudsen's paradigm. The idea that all of the tumor suppressor genes are present in two copies, and even if you inherit one bad copy, like a bad copy of BRCA2 from one of your parents, the other good copy will keep things working all right until it's somehow lost, and only then you get cancer. 2 hits, he called it. And that is indeed how we have envisioned the work of BRCA2. You inherit one bad copy from one of your parents, but the other good copy works until you lose that second copy in certain cells. We don't know how. And when that happens, and it's an event called loss of heterozygosity, LOH. Don't worry about the term, but just remember the abbreviation, please, because you'll see it in some of my slides. Then you get cancer. And our previous work shows you get cancer because when you lose both copies of BRCA2, you can no longer repair your DNA. So you get lots and lots of changes in your DNA, and that drives carcinogenesis. All right? So the story I'm going to share with you starts about 15 years ago. And this was work from Ferdinando Skoulidis, who was at that time an MD, PhD student in my Cambridge laboratory. Nandos now heads thoracic oncology at the MD Anderson Cancer Center. What he did was to build a mouse model for pancreatic cancer connected with BRCA2. So we built at that time the first model that accurately recapitulates all the events that happen in humans in order to be able to study how loss of BRCA2 can cause cancer. Big surprise. Nando's showed you do not need to lose the second copy. You still get cancer in the pancreas in these models, even without loss of that second copy, no LOH. I made the poor man repeat some of these experiments 50 times because I just didn't believe the results, but they were correct. And we were able to go to the Icelandic cancer registry with Jon and Jorunn because many people in Iceland inherit a mutation affecting BRCA2, similar to what we had engineered in the mouse. And they too, many of these pancreatic cancers did not lose the second copy of BRCA2. It's a violation of Knudson's two-hit idea. There was a lot of controversy at the time when we published our results because AstraZeneca was just introducing PARP inhibitors for cancer. PARP inhibitors only work when there is loss of heterozygosity. If the second copy of BRCA2 is not lost, the tumors are resistant to PARP inhibitors and platinum compounds. AstraZeneca was not, we obviously shared the results and not everyone who gets treated with PARP inhibitors, we believe, will be sensitive. This is from the Cancer Genome Atlas project in the National Institutes of Health. And you can see here, they looked at 10,333 cases of cancer in the United States. And again, what you can see from that flagship project is in breast cancer, pancreatic and other cancers, many cases of BRCA2 associated cancer do not lose that second copy. There's no two-hit there. Interestingly, in ovarian cancer, which is highly responsive to PARP inhibitors, almost all of the cases do lose that second copy. We don't understand why the difference between the breast, ovary, and other tissues, but it does help to explain the exquisite sensitivity of ovarian cancer to PARP inhibitors and other compounds that leverage the loss of repair by homologous DNA recombination. So for the last 15 years, I'm afraid we work very slowly in my laboratory. We've been trying to figure out why. And that has led us to a really interesting set of observations that we think can help to explain how the environment cooperates with our genetic background to induce cancer. The first clue came from studies that Sean Tunn, who was a PhD student then in my Cambridge laboratory, did. So as I told you, if you lose one copy of BRCA2, no problem, everything is fine. It's only when you lose the second copy of BRCA2, you get problems leading to the development of cancer. So I asked Sean to take cells which we had genetically engineered to be deficient in one copy or two copies of BRCA2 and throw every compound he could think of onto those cells and ask, was there ever a difference in the way that these cells responded? And the poor man went through literally tens of different compounds with no difference. In other words, the cells with one copy of BRCA2 were just fine. They had normal DNA repair, normal replication protection, no issues, until he threw aldehydes, particularly formaldehyde and acetaldehyde, onto those cells. Now, I think many of you will know, even if you're not scientists, will know about formaldehyde and acetaldehyde. When we drink alcohol, it gets broken down in our body to acetaldehyde. If you inhale car exhaust fumes, you're inhaling formaldehyde. Many of our cosmetics, shampoos, contain formaldehyde. The glue that holds our furniture together contains formaldehyde. Aldehydes are everywhere in our environment. They are also made in cells by endogenous metabolism. For example, some of the processes that control the expression of genes, histone lysine methylation, generate aldehydes as an intermediate product. So this was really interesting to us because it suggested that maybe what was going on was that the aldehydes were doing something to cells which still had one okay copy of BRCA2 that was causing carcinogenesis. And that is indeed what we found. And this effect is not on genes, it's on proteins. I'm going to walk you through the scenario. So here you have a normal cell, 2 copies of BRCA2, and you see they have lots and lots of BRCA2 protein, so they can function just fine. You look at a cell from a mutation-carrying family, 1 correct copy, 1 bad copy, so they have about half the amount of protein. Now we expose them to aldehydes, and what happens is the protein is degraded. We discovered a new pathway that degrades the protein after aldehyde exposure, and then this interesting thing happens. These cells have half the amount of protein. So when that protein is degraded, the level quickly drops between the adequate threshold for function. These cells can no longer repair DNA. They can no longer protect replication. The same thing happens even in otherwise normal cells, but they're less sensitive because they have more BRCA2 protein to start with. Yes? But you can see how a genetic defect with an environmental exposure can actually cause, through this very selective pathway working inside the cells, what we call induced haploinsufficiency. An induced defect can lead to genome instability and potentially to carcinogenesis. So this idea was very striking to us because we wondered whether this might help to explain how people were developing cancer with 1 good copy and 1 bad copy of BRCA2 through environmental exposures. What's the aldehyde or aldehydes that are responsible? Now this is really difficult. There are hundreds of aldehydes in our environment produced inside our bodies. But a clue came from Miriam, who was my penultimate Cambridge PhD student, who was at that time working with Tobias at the Cold Spring Harbor Labs in New York on a totally different idea. We were looking at this phenomenon called cancer cachexia. Many people with cancer just waste away even though they eat. Their bodies just dissolve, and that kills them, and we were trying to understand the basis. And what Miriam found was that aldehydes that were released during the process of cell energy metabolism might actually be responsible for that defect, in this case with a ketogenic starvation diet. So we wondered whether energy metabolism might be linked to cancer in BRCA2 mutation carriers. So I'm going to have to introduce you to another concept, right? We have billions of letters of DNA. We can sequence them at great accuracy and resolution. So if we did this sequentially over time, we can look for changes between DNA, say, today versus how the DNA was 1 month ago. And what Mike Stratton and his colleagues at the Sanger Institute, particularly Ludmil Alexandrov, now at the University of California at San Diego, have found is that in virtually all forms of cancer, there are small changes in our DNA that assemble in patterns which they call single base substitutions. Changes in just 1 or 2 of the letters in our DNA code distributed all across our genomes. These patterns can be classified into over 80 different groups, and virtually all types of cancer have these groups. In fact, Cancer Research UK, just Wednesday, has released a big global challenge. Can we understand how these patterns arise in different forms of cancer? This, for example, at the top, SBS4, is diagnostic of the mutations induced by tobacco in the lung. So these patterns of mutation seem to be linked to changes that drive carcinogenesis. SBS patterns, that's all you need to remember. So we made cells in the laboratory that had 2 good copies of BRCA2, 1 good and 1 bad copy of BRCA2, or 2 bad copies of BRCA2 to mimic what you see during carcinogenesis. And what we asked was, can we treat these cells with aldehydes and induce the early SPS mutagenesis patterns that signal the onset of cancer? And to do that with Andy, who is my PhD student, and Jason, who's my collaborator in Singapore, we assembled a rather monumental DNA sequencing effort where we literally took clones of cells arising from just one single parent, and we followed these cells over long periods of time, months, and asked if you expose them to aldehydes, like the aldehydes you find in energy metabolism, does anything change in their DNA? And I'm going to walk you through this experiment, which mimics what might happen if, for example, you have a poor diet, or you have diabetes, or other alterations in energy metabolism. How would they work on cells? So what I'm showing you here are cells with 2 good copies of BRCA2, 1 good and 1 bad het, or 2 bad copies of BRCA2. And all you have to pay attention to are these blue bars, which show whether we are seeing the SPS patterns that say something is happening, changing in the DNA of these cells that are making them more prone to carcinogenesis. So you can see in cells which have 2 bad copies of BRCA2, this is already there from the beginning. You don't have to expose them to aldehydes. They're already undergoing the mutagenesis that makes them into cancer cells. But it doesn't happen in the heterozygous cells because the 1 good copy of BRCA2 is still working to prevent mutation. But look what happens when we start to add aldehydes generated, as I said, by bad diet, by glycolysis, for example. And you can see over days and weeks, the cells with 1 good and 1 bad copy of BRCA2 are beginning to acquire these patterns. And you can see by the end of this experiment, this mutational signature, the changes in the genes, the changes in DNA are happening in virtually all of the heterozygous clones with 1 good and 1 bad copy of BRCA2, suggesting that this environmental exposure can indeed cause the earliest events associated with cancer formation. But I told you the cells have 1 good and 1 bad copy of BRCA2. So obviously our critics said, maybe what's happening is some of those cells are losing the 2nd good copy. Yes? But we tested that. And what I can show you is that at the level of DNA, at the level of protein, at the level of function, right through to the end of the experiment, that 2nd copy of BRCA2 is still there. It's just that it's being degraded temporarily by exposure to the aldehydes. And the aldehyde we used is called methyl glyoxal. And the physicians in the audience will know that methyl glyoxal is a key intermediate in energy metabolism by glycolysis. When you have high sugar levels in the blood, cells move towards using this form of metabolism rather than their normal metabolism, generating methyl glyoxal, MGO. And methyl glyoxal has been connected with the abnormalities we see in the eye, in the heart, in diabetes, because when you have diabetes and you have high sugar levels, you generate a lot of methyl glyoxal. In fact, a very common clinically used biomarker, HbA1c, is simply hemoglobin plus methyl glyoxal. And what we are showing is that excessive methyl glyoxal can actually cause mutations that can set cells on the earliest steps to cancer. So far, in the laboratory, does this work closer to real life? So here, through the generosity of women who are undergoing surgery to remove normal breast tissue who are carrying the BRCA2 mutations, we were able to develop organoids which recapitulate the cells from these women. And you can see exactly the same thing happens when you expose these cells to methyl glyoxal, the aldehyde. You again see the occurrence of these genomic alterations signifying the advent of cancer. We can see this even in breast cancer tissues. We can see that even in women who have breast cancer without BRCA2 mutations, there's a small fraction which we think may be related to diabetes or metabolic derangements that bring about the mutations that seem to accelerate carcinogenesis. So what does this all mean? Well, we think that we have forced a revision of the 50-year-old idea from Al Knudsen that you need to inactivate 2 copies of tumor suppressor genes to get cancer. What we are showing is that even if the second tumor suppressor gene is still good, that temporarily metabolic alterations potentially induced by environment, exposure to aldehydes, poor diet, diabetes, can cause temporary inactivation of the protein, leading to the changes in DNA that could signify the earliest steps in cancer predisposition. And I think you'll all be aware there are over 500 million people with diabetes, particularly in countries like India. We know that diabetes is connected not just with eye and heart disease, but with an increased risk of cancer. At the moment, theories range from inflammation to high sugar levels promoting cancer growth. What we are suggesting is that there may be another mechanism, which is induction of changes in DNA by this so-called metabolic bypass that we have described that may potentiate carcinogenesis in people with diabetes and other metabolic disorders. Soon after we published this work last year, another group showed that another tumor suppressor, p53, a very famous tumor suppressor, inactivated in over half of all cancers, is again subject to inactivation temporarily by products of energy metabolism, in this case, lactate. So we think that the idea that protein levels are affected by our environment and can trigger the changes that lead to the earliest steps in carcinogenesis may be much more general than just applying to the BRCA2 tumor suppressor. And we wonder whether these types of mechanisms might help to explain why our environment and our genes can collude to cause cancer. So my colleague in London, Charlie Swanton, had looked deeply at cancer genomes and was thinking about how cancer evolves. Think about speciation in a Darwinian sense, right? You have lots of species with different genes. What killed the dinosaurs and led to the rise of the mammals? The idea is that simple linear evolution, step by step, is not enough to explain these big changes. Instead, Charlie has proposed that even in cancer, we may need macro events, like a comet hitting the Earth and wiping out all of the dinosaurs, allowing the mammals to rise. Similar things may be happening in cancer. So we wondered whether mutation due to metabolic changes, poor diet, for example, could be such a macro evolutionary event. And this is an ongoing experiment. We have not published it yet, but we now have these cultures going for nearly a year. We're trying to recapitulate cancer evolution in the test tube. And you can see that when we expose cells to, for example, products of glycolysis like methyl glyoxal, you induce the mutation. You take away the stimulus and the cells are normal. You add back the stimulus and it comes again. But the interesting thing is that if you take away the stimulus, although The signs disappear, the mutations that were caused were still there and can potentially drive cancer. We call this a hit and run episode because you can't see the effects, all of the effects later, but we know that invisibly they are driving cancer. So, to summarize really, we know that obesity, diet, metabolic disorders, many different factors other than our genes are leading to the accumulation of pre-malignant cells causing carcinogenesis. And we think that our new discoveries concerning connections between metabolism may help to explain a link between these very, very common causes of cancer, which we don't understand, and how the earliest genetic changes leading to cancer actually occur in the genome. So I hope in the last few minutes I've been able to tell you about the two questions that my lab has been addressing over the past 20 years or so. A very simple first question: why are some of us more susceptible to cancer than others? And I've taken you through a very recent story which links diabetes, obesity, poor diet to carcinogenesis. But there's another thing that we've also been doing, which Srinivas kindly alluded to, which is what can we do about this, particularly with relation to drug discovery. And I'd like in the last few minutes just to talk through some of our work in the arena of therapeutic lead discovery. You may ask, how is this connected with early carcinogenesis? My belief is that the current armamentarium of drugs used to treat cancer is not going to be enough for early intervention. We need safer, better tolerated, new entities working through totally novel mechanisms. So we have devoted a lot of effort over the last 10 years in my laboratory to pulling together genetics, structural biology, chemistry, and imaging to try to address three problems. How do you find the best new targets? Once you find the best new targets, how do you drug them if they've never been drugged before, if they are not kinases, if they are not G protein coupled receptors? And finally, how can you actually see how these new drugs may work in cells so that you can develop biomarkers to introduce them into the clinic? So let me talk you through just one of the problems. I think, as many of you in the pharmaceutical industry will know, the industry has been very successful at classical druggable targets. We are able to drug protein kinases, G protein coupled receptors very effectively. I don't think academic research has much to contribute. Similarly, there are so many ways of making antibodies. What I'd like to introduce you to is this. Most things that are going on in cells are not just through kinases or G protein coupled receptors. They are through the interactions between proteins and nucleic acids, macromolecular interactions. There are potentially hundreds of thousands of these that regulate every cell, and currently they're an untapped and relatively inaccessible class of drug targets in industry. So how does one identify and drug this new untapped class of interactions? So this is the new technology that we developed in my laboratory. And the idea harnesses evolution. Think about it. As I said before, many of the proteins that are working in our bodies have mimics in the simplest organisms, bacteria, for example, which thrive in all sorts of different harsh environments. So we decided some years ago to test the idea that if we harvested proteins from very simple organisms and introduced them into human cells, they would serve as probes for shapes. Rather than using chemical libraries, we decided to use protein-based libraries. Why did we do this, you may ask? Well, the pharmaceutical chemists often talk to me about chemical space, this vast entity, 10 to the 63 and such numbers. Professor Mehta is smiling because I'm sure he's familiar with these arguments. We wondered how much of that synthetic chemical space is actually relevant to human biology. I think that human biology occupies a relatively small proportion of that space, and thereby biologically inspired shapes like we derive from simple organisms may better represent that chemical space than the full synthetic repertoire. And so what we do is we introduce these shapes. We have libraries now of over 10 to the 9 to 10 to the 12 shapes derived from genetically encoded things derived from bacteria, archaea, yeast. We introduce them into cells. Like chemicals, they go and bind to intracellular proteins and induce phenotypes. They may kill a cancer cell. They may make a diabetic cell secrete insulin. But we have a little tag at the end of each one of these probes so that we can pull it out when it causes an effect and identify the target to which it binds and the structure of that target, which means we know very quickly whether it's druggable, whether it's ligandable, and it gives us the template to make new drugs. So this process, which is called site seeker, because it seeks sites, has been patented and has led in Cambridge to the spin-out of Foremost, which is dedicated to drugging the undruggable. And I'm pleased to say that this work, some of which we published a few years ago, has led to many big pharma companies now taking up this technology to identify new targets for their own pharmaceutical research. Foremost itself now focuses on finding new glues for this very exciting area of protein degradation and PROTACs, but the technology is being used by many big pharma companies to identify targets for their own pharmaceutical programs. We have also been trying to drug the undruggable, developing new chemical libraries, new ways of using fragment-based chemical discovery, all guided by X-ray structure, structural predictions, considerably facilitated now with the advent of AlphaFold and other such AI algorithms. But here, and some of this has been done in the center in Bangalore, we have so many new, previously undrugged targets where we are able to generate new drugs, some of which, this one is just moving into pre-IND now. We have licensed it to industry, and we think it can be used to target currently intractable RAS-dependent cancers, for example. And last but not least, my work with Alessandro, who is still back in the UK. I'm not going to take you through it, but to be able to find pharmacodynamic markers of activity of these new types of inhibitor, we've developed what we sometimes call hyperdimensional imaging microscopes. We built new microscopes that can interrogate not just classical elements of photophysics, spectrum, fluorescence, lifetime, rotational anisotropy, but can now capture all of those aspects of photophysics in a single detector in real time. And so we can deconvolute multiple signals which are being perturbed by the action of these drugs to identify the mechanism of action. So I hope I've told you how the work on disruptive technology platforms to facilitate lead discovery has also been commercialized, as Srinivas alluded to, through companies that have spun out from my laboratory. And I hope now that you can see why I have great hope that as we develop safer, better tolerated mechanism targeting agents, that we will reach the dream of being able to intervene early in cancer, long before it becomes a clinically significant disease, and in the interim, to actually make treatment itself much more efficacious. So I will stop there and simply give you a plug for those of you who are interested. I met Dr. Reddy here in Hyderabad in 2011 when I was embarking on this journey to try to do some of this work in India. And he was totally inspiring, very positive about how it is possible, despite all the difficulties, to do things. And with generous support from INSTEM, NCBS, Department of Biotechnology, and other sponsors, most recently the Gates Foundation, the CCBT is operating as a multidisciplinary research center. We're working with the Gates Foundation on some of their new drug discovery programs for low middle income countries like India. And I've also been able to work with a fantastic bunch of quantitative engineering colleagues at the Indian Institute of Technology in Madras to develop the Center for Integrative Biology and Systems Medicine, the idea being to bring the quantitative sciences to human biology. And they've had some wonderful breakthroughs ranging from the design of new influenza vaccines to cataloging the Indian genome to finding, using AI, markers that predict preterm pregnancies before the children are born so that these women can be monitored very cheaply and very effectively. So I'm going to stop there and just say my thanks to all of the support I've had from the Medical Research Council, the University of Cambridge, my current institutions that I work at in Singapore, and my laboratory. We do have fun from time to time, and when I'm not working in the lab, I like to be out climbing mountains. So thank you very much for your attention.
Thank you, Professor Venkatraman, for a truly illuminating lecture. Just want to say that there is a lot of silo thinking when we come to cancer, and what this entire story that you have woven, and eventually showing a very strong connect with inflammation and diabetes, and how this can really induce, and at the same time nailing down the certain environmental factors. I think you have kind of covered not just the prevention, but also a kind of sensitization about how one can essentially keep themselves hopefully away from these mechanisms that are overridden. With that, I'm sure there would be questions.
Sure, I'm very happy to take questions. I hope I was able to convey a sense of my excitement. I see BJ has his hand up.
I have one question and one comment. Comment first. The genome stability is maintained by a plethora of very important proteins. BRCA2, BRCA1 are some of those. There are many more.
Of course.
Are you suggesting that we have just discovered BRCA2 as a marker of instability, mutation? There are others which we have not discovered? Or these are the ones and these are the only ones? This is number one comment.
Sure. May I answer that?
Please, please.
So I think your point is well taken. Clearly, as you know, there are at the moment over 50 proteins that have been implicated in double-strand break repair by homologous recombination, probably about 20 involved in non-homologous end joining, and a plethora of proteins involved in different repair processes. So clearly there are more. What's really intriguing is, after our work was published, one paper before, some other members of these families, like MR11 endonuclease, have also been shown to be conjugated by small molecule metabolites generated during energy metabolism. So whilst the idea of metabolic bypass and the impact of metabolism on DNA may be far beyond just BRCA2, I think that this will be a much more general mechanism than just BRCA2. And where it extends, we don't know. The reason I'm very excited about this idea is that for a long time, we have thought about these small molecule metabolites as byproducts, for example, of energy metabolism. We haven't really understood how they cause pathogenesis, whether it's in diabetes. How do you get the microvascular changes? How do you get the risk of cancer? And I think this type of work is beginning to show that some of these metabolic disorders don't just affect metabolism, they can affect DNA through the types of effects that we have just demonstrated. And I think that's a very interesting new area that might be quite fruitful to investigate, both for pharmaceutical targets as well as for just understanding how these diseases, chronic diseases, arise.
Yeah, the second one, you didn't elaborate too much on this. The circulating DNA is a very good giveaway. If we can read it well early enough and very sensitively enough, I think it will tell us a lot. Do you agree with that? Are we doing anything about it?
Yeah. So that's another very good point. So tumors secrete, if you will, small amounts of DNA into the circulation. You can sample peripheral blood and you can ask, do any of the gene mutations that drive cancer, are they present? And the frequency seems to be related to whether you have early preclinical disease or not. And many companies, including Grail in the United States, have also introduced the concept of looking at methylation patterns to be able to even begin to nail down tissues from which these cancers can arise. The utility of these tests is impeded by two things. One is, if you screen normal, healthy people first, will they consent to the screening? They don't have cancer, they're happy, they don't want to necessarily know for insurance reasons, for employment reasons. The second thing is, all of these tests so far give an index of suspicion. They don't tell you definitely have cancer. They say, if you have this DNA, you may have cancer. And often they don't tell you exactly in which tissue. So the follow-up has to involve imaging, usually MRI-based imaging, which is extremely expensive, particularly if you have to scan multiple parenchymal organs. So there's a cost issue. So as a result of this, Grail has embarked on a study which is about to wrap up in the NHS in the UK, where they have looked at about 350,000 normal, healthy people in the older age group to see how many of them have this high index of suspicion, and about 150,000 higher risk individuals to see if the signal is elevated. One of the problems with any of these primary prevention studies in the population is the number of events. If you take 100,000 people, in 2 years, 3 years, 4 years, how many of them are expected to get cancer? Not that many. So some of these studies take a lot of time, and there are impediments in terms of cost effectiveness. So my own view really is, while these tests have their value, probably the lowest hanging fruit is high risk populations. Smokers, there's clear evidence. Potentially, we think, through some of the work I've described, people with diabetes or metabolic disorders may be another very common high-risk group. So I think there's a lot of work still to be done about risk stratification before we begin to apply these tests.
So I enjoyed your presentation very
Thank
much.
you.
So I think you nicely articulated that phenotypic screening can throw out some really novel targets. A couple of questions. In your peptide-based screening, did you come across cases where more than one peptide, more than one target give you therapeutic benefit? That's one question. Second, instead of peptides, have you also looked at metabolites? Instead of making the cancer worse or making worsening the carcinogenesis, can they also give you the benefits?
Those are both great questions, Murali. Thank you. Let me take each of them in turn. The first is, please remember that we are not advocating peptide-based therapies. For us, the peptides are a way to identify the binding site and get the chemistry right to create small molecules. Yep. So when we screen, we often find, even in the primary phenotypic screen, we get more than one hit. But what's really interesting is once we reduce it to 1, 2, 3 targets, we can do further in vitro screening, and then we can expand the hits which bind potentially to the same site. You can see how we can easily use displacement assays to see whether they bind to the same site or not. And some of these may not just be potential binding sites for inhibitors or activators, they can also be binding sites for PROTACs because they are selective and they can really degrade targets, for example. So we think that this type of approach to identify binding sites which are involved in phenotypes can really lead to more first-in-class drugs rather than the fast follower types, which could really lead to different benefit. So I hope that addresses your questions, but please tell me if it hasn't. The metabolites is another very interesting problem. So we have recently in Singapore developed new ways of exploring in vitro some of the targets that we get to try to characterize new binding sites. So with Zachary Gates, who is a postdoctoral colleague in Singapore, we've developed new ways of including non-proteogenic chemistry, some of which include these sort of tertiary modifications on the amino acid side chains to explore different chemical space, some of which could be influenced by metabolite-dependent conjugates. It's looking interesting, but it's still very early. The techniques are in development, but again, we hope that this will help us to identify new binding sites, but also potentially new chemistries which engage even known binding sites. to give us better compounds for further progression.
Thank you, Professor Venkatraman. For the interest of time, we can maybe discuss with Professor Venkatraman during the lunch.
Thank you very much once again.
Thank you, Professor Venkatraman. May I request Mrs. Anuradha to present a souvenir to our speakers, please?
Thank you. Thank you so much. It's a real honor, and I'm very grateful once again to your family for allowing me to speak here at this really impressive event. Thank you. Oh my goodness. Thank you. Thank you very much. Thank you very much, Srinivas.
It is now an honor for me to introduce our next speaker, the founder of LV Prasad Eye Institute, Dr. Gulapalli Nageshwar Rao. Dr. Rao received his MBBS degree from the Guntur Medical College in Andhra Pradesh, followed by medical training in ophthalmology at the All India Institute of Medical Sciences and the Tufts University School of Medicine, Boston. Mr. Prasad has, in the morning, alluded to some of the very important contributions of Dr. G.N. Rao. I will not go to the entire details, but let me again mention about amongst the several international and national awards that were conferred to Dr. Rao. One of the most, something that we all cherish, is his induction into the Ophthalmology Hall of Fame at the meeting of American Society of Cataract and Refractive Surgery. He was conferred by the Government of India the 4th highest civilian award, Padma Shri, in 2002. Again, as Mr. Prasad mentioned, Dr. G.N. Rao has made an impact on people from all strata of the society. This is something which has been a game changer in the way therapies and access to medical health and cures can be made available, delivering quality eye care in an affordable manner, and making LV Prasad Eye Institute truly an iconic model for the rest of the world to emulate. With this, sir, we look forward to your experiences of having interacted with Dr. Anji Reddy and sharing those with us. Thank you, sir.
Satish, Prasad, Professor Venkatraman, members of the Dr. Anji Reddy family. At the very outset, our deepest condolences to the family on the loss of Mrs. Reddy on behalf of our institute. Remember, Anu and Satish should remember, you both have become a little sharper because of the Krishna district genes you acquired from her, you inherited from her. I happen to be from Krishna districts, that's why I'm making the claim. Last May, I was at the Mandarin Oriental Hotel in Paris. Flipping through the hotel magazine, I was struck by one story, the story on Araku Cafe. Where is Araku? Where is Paris? Araku represents one of the most marginalized communities of the world. People totally disadvantaged by geography, and economy. Paris represents the other end of the spectrum: fashion, luxury, and sophistication. And the connect was made by the specialty of Araku Valley coffee. So then I became curious. I called Manoj, the chief executive officer of Nandi Foundation, to tell me the story, how it all came about. I believe he and Dr. Reddy used to go on morning rounds every day, morning walk. And one day Dr. Reddy asked him, after the founding of the Nandi Foundation, let's not take small challenges. Let's go around town, meeting some prominent people, and ask them for some of the biggest challenges that we have in our society. And let's pick one of them and go after it. And I believe Mr. Sankaran, who was the director of ASCII at that time, told them, if you are really looking for a challenge, go to Araku Valley and transform it. And it's immediately piqued the interest of Dr. Anji Reddy, because he was a man who was always looking for audacious goals. So he was asked to go and visit Araku Valley. Manoj went there, studied the place, and came back and said, it's completely devastated, a lot of poverty, no health care, no water supply, no education. But the one problem that he picked initially was the very high degree of maternal mortality. So they targeted that first. And from there, it extended to all forms of primary health care, then to education, then to agriculture. And in agriculture, they thought, how do we start? And they again talked to the local people and heard from them, historically, Araku was a place where coffee was grown by the Britishers in the early 1900s. So they decided, why not go again to cultivate coffee? So that's how the whole story of Araku coffee started. Then it became an international brand, and that's how it found a place in the magazine of a sophisticated hotel in Paris. The second story that I heard, what made Dr. Anji Reddy different, in which I also believe, is if you go anywhere, keep your eyes and ears open, you can learn a lot of things. One example was, apparently he was staying at the Oberoi Hotel in Delhi once. And in his hotel room toilet, he found a sign that the water coming here is potable, but if you still want mineral water for your consumption, we'll be happy to provide it. So he came back and said, if Oberoi Hotel can provide potable water in its toilets, why is it that we are not able to provide drinking water to all our populations? That started his new project on water, to supply many communities with potable water, drinking water. Because India's one of the biggest challenges is the supply of drinking water to all our population. And I believe now in many of the corporate headquarters of Hyderabad also, the same group is supplying their drinking water. And the third time that I heard got involved with Dr. Reddy was with the development of NICE hospital. Dr. Padmanabha Reddy, founder, is sitting here. And one day, Dr. Reddy called me and said, I'm bringing somebody to meet you. This young fellow is very much involved in neonatal care and wants to work towards taking this care to the most disadvantaged people in our community. And we want to start a new, modern neonatal hospital in Hyderabad, and we would like to discuss with you. They came, they talked, they told me all about this hospital and how it's going to affect the communities, both here and outside. And he insisted that I should become a member of that board. I did not contribute much. I was there on the board for a few years, but Padmanabh went on to develop a world-class facility. And more importantly, from my point of view, it's not what he does in Hyderabad in the nice hospital, but what he went out and started doing in multiple remote communities of India and transforming lives of thousands of people and touching millions of families. And then the story with us at the L.V. Prasad Eye Institute. Reddy's Labs was started in 1984. We started our institute in 1987. He always used to tease me. We have started about the same time. Now I have made 100 crores. What about you? So I used to tell him, you made 100 crore rupees. My aim is to treat 100 crore people and eliminate blindness in 100 crore people. So he was very appreciative of that. So the first thing I heard about him, and I heard he believed in giving. So I called his office and asked for an appointment, I think in the early 90s sometime. So he was very considerate, gave me an appointment immediately. And when I met him, he said, I heard about you and your institute and the excellent work that the institute is doing, particularly for the poor, and how can I help? And I told him that we needed support to continue our work. And he immediately said yes, and that was for supporting cataract surgery in the underprivileged people on the name of his father, an endowment to support such surgeries for a period of 10 years. So that was the first connection. Then he started asking me, are you doing any research? I said, yes. Research is a component of our institute, very active component. And at that point in time, we were just about to launch our molecular genetics laboratory. And he asked me, what is it that you need to make it better and carry your work faster? And I told him of the need for more equipment in the laboratory. And he immediately said, get it, and I'll support it. With his support, we started the molecular genetics laboratory. And just 4 years ago, for the first time, when global ranking was done for every specialty research, and when we were ranked among the top 10 eye research laboratories in the world, I missed the presence of Dr. Anji Reddy, because he would have been thrilled to hear those news, because he was such a support. Every single time he came to meet me, he used to call me and say, are you free for a cup of coffee with me? He used to come over, sit across from me, and then talk about research and research and research. That was his passion. And he used to keep on asking me, what more are you doing? What more are you thinking? And a couple of years later, Balu explained to him, took him to the lab and showed him around. And then when Balu told him, there were some pieces missing. His immediate reaction was, why are they missing? Get them. And that's how the lab was further strengthened. And then he got onto our board in the early 2000s. I think Govardhan Mehta and Dr. Anji Reddy were on our board together. And one day we were discussing the future of the institute long term. And the discussion centered around the raising an endowment and creating an endowment for the long-term sustainability of the institute. And immediately he said, that's not a big amount. 10 people together can give it. And I told him, you make it sound so simple. I need your help then to get those 10 people. The very next day he called me and said, can I come and see you? He came, he gave me a small handwritten note, and actually we photographed and kept it in my office even till today, and Satish has seen it and taken a photograph of it. And I said, this is the first installment of my contribution, and I'm going to talk to others. And he did, and he convinced a few others to support us. So in many different aspects of the growth of LV Prasad Eye Institute, Dr. Anji Reddy played a pivotal role. It's because of that today we have a record of touching the lives of nearly 400 million people. We have a record of touching 20,000 eye care professionals around the world through education, published over 5,000 peer-reviewed papers in research, and touched 200,000 irreversibly blind people through rehabilitation programs, and became the biggest eye bank in Asia-Pacific region and the biggest corneal transplantation center in the world, in a country where people told me organ donation is impossible and don't think of transplantation. So it is people like Dr. Anji Reddy that made that possible. When you see those three, he contributed to the advancement of science in our institute and made it fit for the definition of the word institute. When we think of institute, I think the definition for us is creating new knowledge, disseminating new knowledge, and practicing new knowledge. And Dr. Anji Reddy certainly contributed to all those three at our institute. The second is society. Today, we are in 308 locations in 3 states, and 280 of them are primary care eye centers in the remote, rural, and tribal communities. Through that, we are making real contribution to the society. And that's where he contributed. And then sustainability. We follow all the principles of sustainability, green energy, trees, carbon print, everything is taken care of through various efforts that are going on in our institute. So in some sense, we have satisfied the criteria of what Dr. Anji Reddy stands for. Some years ago, I ran into an adage which said, go into the world and do well. More importantly, go into the world and do good. And Dr. Anji Reddy's life reflects this so admirably. And today, if you go on road number 2 of Banjara Hills, you see the campus eponymous with Dr. Anji Reddy, our campus. And that's where all activities of LV Prasad Eye Institute happen. It's the headquarters. And It's a manifestation of what can be done in the field of healthcare and how healthcare can be provided to everyone that needs it at the same quality, defining the very concept of universal healthcare. So we are always very grateful for him and remember him on many occasions. And every day, as we pass through the corridors, we also see many areas with his name on it. In fact, the head of our campus is the Kallam Anji Reddy Distinguished Chair of Ophthalmology. And then we go down, we see the molecular genetics lab on his name. So through various areas of our institute and the activities, Dr. Anji Reddy made a remarkable impact on us. So again, I remembered of one of Mahatma Gandhi's sayings, and he certainly qualifies for it. Strength does not come from physical capacity. It comes from an indomitable will. And Dr. Reddy has that in ample measure in his life. That's all I want to share with you. This is not as exciting as the exciting frontiers of biology that Professor Venkatraman has taken you to. But I think at least I have shared a few facets of the wonderful life of Dr. Anji Reddy and how he touched and transformed the lives of millions of people, both through the industry and his giving. Because as far as pharmaceutical industry is concerned, he laid the road and the entire world walked on that road to reach Hyderabad, the entire pharmaceutical world. So he made it one of the epicenters of pharmaceutical industry in the world. And his contribution in that sector cannot be forgotten. So that combination of innovation, science, research, and his intense sense of responsibility to society are unmatched. For all that, we'll have to continue to remember people like him, and may the younger generations take inspiration from such wonderful examples in our society. Thank you so much.
Thank you, Dr. Rao. May I request Mrs. Deepthi Reddy to present a souvenir, please? Can we have Dr. Rao on the stage back, please? Thank you. May I now request Dr. Anji Reddy? I would like to now request Mr. Satish Reddy, Chairman, Dr. Reddy's Laboratories, to share his concluding remarks for the day.
So it's almost noon, so very good afternoon to everybody. Delighted to have all of you with us here today at the third Dr. Anji Reddy Memorial Lecture. First wanted to thank Professor Venkatraman for delivering the lecture. It was quite fascinating to hear. It also puts a lot of things to me personally in perspective. As you have heard, I lost both my parents, my mother very recently, and both of them were victims of cancer. So it kind of puts it in a very personal perspective, because when it was diagnosed in both their cases, the first thought that comes to me is, and also the family, is, why them? Why did it happen to them? So could it be this? Could it be that? Could it be genetics? Could it be, anything, which had to do with their lifestyle? So these kind of things keep coming into the mind. but then to kind of process it and also see it in perspective from what you put at this point of time, and the work still continues, to get more and more answers, and ultimately, hopefully, more and more cures, so that, such great people, we shouldn't be losing them. So that's what was running through my mind when you delivered the lecture. It was a very fascinating lecture, just to also understand the whole genesis of what could be the cause, and the kind of findings that you elaborated in a very lucid and very fascinating way gives all of us hope. Because today, looking at the prevalence, I think when we were discussing, you're also mentioning that it's also a time when there's a younger generation also developing these cancers, which also has happened, because we always thought it was something which comes during age, but the kind of statistics that you also mentioned, it is a serious thing. So to also understand it in perspective, and especially we as a pharmaceutical company, as you can see, Murli, who is here, and also you've witnessed Origin, the facility, we are working seriously in that field, and that elusive dream, that unfinished agenda which Dr. Reddy had, hopefully we should be able to fulfill it at some point of time, at least during our lifetime. I think that's the goal all of us work with us. So I once again thank you for delivering the lecture. Dr. Rao Garu, it's always a pleasure listening to you recounting memories of my father very recently. In fact, I must tell you my personal experience at his institute. Probably most of the times you won't know it, but because of an eye injury which I had almost about 5 years back, I regularly have to visit his institute. It's one of those rare institutes where you don't have to pull any strings, to get any special treatment. I just walk in, take the appointment and walk in. It's a world-class institute. The efficiency right from the time you walk in till you walk out, and the personal attention that you get from the team that he has built in this institution will always be there with me. So it's also quite nostalgic for me, because every time I pass through the campus that he talked about, the L.V. Prasad Eye Institute, on road number 2, named as Kallam Anji Reddy campus. A lot of people keep asking me, saying that why was it named after Dr. Reddy? And then it shows, from all that he explained about how passionate Dr. Reddy was about research, about institution building, about serving the society, all the traits, as we have heard about L.V. Prasad Eye Institute from the founder himself, they're both very common. And that's why I think Dr. Reddy and Dr. Rao shared a very, very deep bond amongst them. And recently also when I visited him after my treatment, at his institute, I also took that photograph. I also took the photograph, Dr. Rao, of this Molecular Genetics Institute, to see how it has flourished. And I was so happy to hear that it's been ranked among, one of the leading research institutes. And that's something I think, especially when you have a primary objective of serving the society, it shows the institutions that you have built. It shows the institutions Dr. Reddy has built. We also talked about a nice foundation. Dr. Padmanabh is here. These are the kind of institutions which I think are here to stay. They inspire a lot of us. They inspire several people that we can also serve the society and keep it as an objective. It's not just about making money. I also wanted to acknowledge the presence of prominent people in this audience today, people who have known Dr. Reddy very closely, Dr. Govardhan Mehta especially, has known him very closely. In the past couple of times also, he has spoken about his deep association with Dr. Reddy. He also reminded me this morning that he knew him much before us, right? So it's been a very, very long association that goes back to NCL Pune. I was not even born at that time, so that's what he was referring to. Also wanted to thank the team which has organized this event, starting with Srinivas Suriganti. He takes very personal interest besides the duties that he discharges as, that's Srinivas there. So he, besides his duties as the director of the Life Sciences Institute, Dr. Reddy's Life Sciences Institute, takes personal attention and makes sure that everything goes according to plan. So thank you, Srinivas, once again for this lecture. Also people in my team, Dinesh, Usha, Raju, several others, who made this event possible, taking care of logistics, taking care of a lot of arrangements. Also, I wanted to talk briefly about this roundtable we started just last year. In fact, last year when we started, it was a roundtable on innovation. It was a subject very close to Dr. Reddy. And it was about emerging frameworks of innovation. As you can see, there's a lot that needs to be done, especially in India, to encourage innovation. There are people in the private sector who have taken it upon themselves. Because as you know, most of the companies here are API-based companies or started with APIs, gone on to make their mark in generics, very highly competitive industry. And then you don't find examples where a generics company would invest into innovation. This is not something that's seen anywhere. But then Dr. Reddy, for example, had this conviction, not just now, but 30 years back when he started the Dr. Reddy's Research Foundation with the prime objective of discovering a drug. And that's an institution which continues to do this work, and that's why I talked about the unfinished agenda. But then it is this kind of a conviction, and we keep constantly talking about how the government also has to pitch in. And that again, Dr. Venkatraman, I hope to learn a lot from, especially Singapore, where you've been closely involved with in terms of creating that kind of ecosystem which can actually help this country also. And that's something we, all of us, look forward. So that was a roundtable which we started with, where people from diverse fields with diverse expertise were able to make their contribution, in terms of bringing out that white paper, which we had disseminated widely, not just in the Script magazine, which is read worldwide, but also circulating it to policymakers and also people across the industry to also see what can be done to do that. And of late, you've been hearing a lot of good news on this front, because the government has also started, to put in money, which hopefully will be utilized to create that ecosystem. Again, it's not easy to do that, as Radha Rangarajan here can attest to, because she heads the director. She's the director of the CSR in Lucknow. Not a very easy thing, because especially when it comes to execution, the administrative difficulties they go through when they need to focus on the research. Also, pitching in from the government to also promote collaboration, for example, is something all of us look forward to. So that was a roundtable which happened the first time. The second time, Dr. Ashok, you were also part of it. This time it was on public health. The public health, especially the theme that we announced this year was prevention, cure, and quality of life. A multi-stakeholder perspective in public health. That was a roundtable. Again, we had eminent people again from diverse fields. From the medical institutions, we had Dr. Devi Shetty of Narayana Hrudayalaya, also Dr. Ajay Kumar, HCG, Cancer Institutes. And we also had people representing philanthropic organizations like the BMGF, we had CIFF, also PAN, various Aga Khan Foundation, people who have done immense work in the public health. They also participated in the roundtable. And we also had people from the government, Dr. Y.K. Gupta, who also is the director of, president of AIIMS Institute of AIIMS Bhopal as well as Jammu, and also in several government committees. We also had Dr. Soumya Swaminathan, many of you are familiar with her. She was in the WHO recently and also involved with the government in various forms. So Dr. Ashok also was participated in that in public health. So a lot of contribution, in terms of ideas, insights which came in. Again, it will come out in a white paper, which we will also make it public and also disseminate it. And that was something which worked out really well. Again, when it comes to Dr. Anji Reddy himself, when it comes to public health, mainly through the company itself, he was very, very, it was a subject very close to his heart. As Prasad mentioned in his opening remarks, access, affordability, innovation. This was a prime movers of what he did in life, and especially when it comes to healthcare, he strongly believed in these aspects, and especially from a pharmaceutical side, I still remember conversations, again, before I joined the company. That was the era of when the fluoroquinolones were just coming in. And just to give an example of how his thinking used to be in those days, in those times when he was in the process of building the company, norfloxacin, which used to be priced at 8 rupees per tablet. Again, we are talking about, 1988 or so at that point of time. And he would talk to people in the company, and he would narrate these stories over dinner time, where he used to say that if only that price can be reduced by half, it can reach many numbers of people. And then there used to be these calculations on the back of an envelope calculations, which on several other molecules, I and Prasad had the fortune of, seeing those kind of ideas, where he would just drop this thing of saying, if it was costing a certain amount of rupees for making the API, the question is, what margin can we retain as the manufacturers keep running the company. But then when it came to norfloxacin, he also calculated at what price it can be provided to the people of India so that many people can afford it and it can reach millions, and how much of tons of API we should produce. So that it was that level of detail that he used to calculate, and that was the reality of norfloxacin. He never talked in all these conversations anything about how much profit we can make. It was all about how many people we can reach and how we can make that happen. And he was also always inspired by this quote from George Merck. George Merck, as most of you know, he was the son of the founder of Merck, and he was the CEO of the iconic company called Merck. And let me just repeat that quote here. And the quote goes as follows: "We try to remember that medicine is for the patient. We try never to forget that medicine is for the people. It is not for the profits. The profits follow, and if we have remembered that, they have never failed to appear. The better we have remembered it, the larger they have been." End of quote. So this was his inspiration, and how he practiced it through his life in terms of bringing affordable, innovative medicines to populations not only in India but across the world. That continues to inspire us. It will continue to inspire us even as we innovate to bring out original discoveries, and that's the unfinished agenda which we still pursue. The institution that he has built, the beliefs, the principles, the values that he has instilled in all of us, that will continue to inspire us. It will continue to inspire generations of scientists, entrepreneurs, people across the country to serve the society, and that's what we look for. And even in terms of giving back to the society, the institutions that he supported, whether it's the Dr.Reddy's Foundation, also the NICE Foundation, for example, they work primarily on several areas, Dr.Reddy's Foundation, but we also work in the primary healthcare services, also in the NICE Foundation, which we support certain programs. And this is something we will continue because it involves upgrading primary healthcare centers, and also the excellent institution Dr. Reddy built up through the NICE Foundation, which is a leader in terms of neonatal foundation and also in terms of mother and child healthcare. We will continue to keep following his legacy in these areas. And once again, I wanted to thank all of you for being here today, for being present here today to commemorate the 12th year of passing of Dr. Reddy. And again, I go back to what Prasad said in the beginning: if only he was here, to have heard scientists, to have heard the memorial lecture, to have heard the institutions which he supported in terms of where they have now come in terms of making the impact in the world, he would have been so pleased. So we once again remember him very fondly on this occasion. And once again, thank you everybody for being here. Thank you.
Thank you, Mr. Satish Reddy. I would like to thank speakers for the day, Professor Venkatraman and Dr. Rao, as well as all the esteemed guests for participating in this memorable event. Thank you all once again, and I request everyone to join us for lunch in the lobby. Thank you and have a wonderful day.